Thank you, Nicole, for the generous introduction. Thank you all for being here. I'm really excited to talk to you all today about some things I've been thinking about recently, and to get your thoughts and feedback, and some maybe discussion at the end about ways to move forward. The question I'd like the question I'd like to ask today is, if we combine genomics with geology and ecology, can that inform us about the predictability of evolution? And this question is evolution predictable? Is one of the kind of long-standing debates, and to what degree is evolution predictable? One of the long-standing debates in biology. So before I do that, though, because as Nicole said, I'm relatively new here again to UH Manoa, and I haven't met all of you, I'd like to introduce myself a little bit. And by way of doing so, introduce some of the concepts I want to talk about today. First, I'll just give a little bit of my own life history. I moved to Hawaii when I was 14, the summer between my 8th and 9th grade year. I attended Kapa'a High School. Some number of years later, as Nicole mentioned, I moved here to UH Manoa, where I was a graduate student in the then zoology department, and I think I was the only student working on plants, but also fishes at the time. And I'm going to warn you guys that I'm going to give you a mostly plant-focused talk with a little bit of microbes sprinkled in, but Nicole assured me that we're an open-minded group and that plants are welcome here. Nicole mentioned my PhD advisor, Tab Fukami, who's a microbial ecologist, left kind of mid-swing in my PhD, and I made the choice to follow him to California. Spent a number of years there, but then I was very fortunate in 2016 to land a faculty position in the biology department at UH Hilo. Had a really fantastic 8.5 years there, part of the TCBS, the Tropical Conservation Biology Environmental Science Master's Program, and Department of Biology. As you can see here, my own life history very clearly follows progression rule dynamics like any good biologist in Hawaii. I'm going to dig into that more in just a minute. But I recently undergone a back dispersal event from Hawaii Island to Oahu, where I joined the School of Life Sciences, particularly kind of on the botany side of things. Part of the graduate program in ecology, evolution, conservation biology, as well as botany. And I'm really honored and excited to be a part of cooperating faculty in your group in C-MΑIKI. Also part of my responsibility as the director of the rock herbarium. I'm not going to talk about that too much today, but if you have questions, please grab me after, shoot me an email. I'd be happy to show you the herbarium. Or if you want to get involved there, just let me know. So in terms of overview, what I'd like to do is address the following things. First of all, is evolution predictable? And I'm going to make a big mistake and try and talk about 3 different concepts of predictability and evolution. I'm going to talk about that progression rule that we just saw. And I'm going to talk about the island syndrome as well as the taxon cycle. So 3 influential ideas about the possibility of predictability. Then I'm going to move into general patterns of endemism in island plants and how they compare to other things like mushrooms, lichens, etc. And really talk a bit about the lack of genetic diversity in native Hawaiian plant lineages and what that means in terms of being able to do this kind of work, the challenges that come with that, and some of hopefully the strides that we're making to overcome some of those challenges. And that leads into the third, which is I'm going to talk a bit about the progress that we've been making and originally kind of phylogenetic to now phylogenomic methods to reconstruct the evolutionary history of Asteraceae. This is the daisy or sunflower family on islands, Hawaii and elsewhere. I'm going to switch gears a little bit to kind of dig into ecology and talk a bit about the evolution of dispersal traits in the Asteraceae, and then come back to genomics and just mention briefly some of the work that we're starting to do now in terms of developing high quality reference genomes and start to do some comparative genomics, and we're moving into a little bit of functional genomics as well. And then ultimately kind of open it up for discussion. I'll mention at the end a new project that I've started since moving here to UH Manoa, that I'm very, very excited about, and it's gaining some steam and some traction, and hopefully it'll really get some wind under its wings, but curious to hear all of your thoughts. So let's jump into it. Is evolution predictable? So as we all know in this audience, the Hawaiian Islands are formed over a magma hotspot as the Pacific plate drifts off to the northwest, just about exactly at the rate that your fingernail grows. And so for the main Hawaiian Islands, we're talking about 5 million years of evolution. So 5 million years since Kauai was where the big island is now, and the magma hotspot is currently feeding Mauna Loa, Kilauea, and Loihi. As the islands move off the hotspot, they subside quite a lot under their own weight. You can see that in this kind of false color image, the kind of deep trench around Hawaii Island and Maui Nui because of their mass. They erode readily. Basalt is something that really breaks down easily, especially at the forces of water. And we have these major landslides that we can see, for example, the Nu'uanu debris field off the coast of Oahu is thought to have created a 500-foot tsunami that circled the entire Pacific. So one of the really influential pieces of work actually came out of a meeting that was held here at UH Manoa, published and edited by Warren Wagner and Vicky Funk of the Smithsonian, a book that was titled Hawaiian Biogeography, Evolution on a Hotspot Archipelago. And in this book, they hypothesize that there may be general rules to how speciation works in Hawaii, and that the geologic history, this well understood geologic history, may make some of the evolutionary outcomes predictable up front, a priori. So what would progression rule look like if we found it in a phylogeny? In a simple progression, we would see something like this, where Kauai are the kind of basal lineages are on Kauai, and then there's dispersal and speciation events down the island chain as new islands arise and suitable habitat becomes available. That's kind of a simple pattern. This is also progression rule. This is a more complex pattern that, as we know, biology sometimes is noisy. So sometimes you get grades and clades, and sometimes you get back migration. But overall, you see the same thing here, a progression rule down the island chain as new islands arise. Speciating away from older islands, as well as speciating, at least sometimes speciating within islands. So this is something, though, that going back to the work of Tuzo Wilson in 1963, it has been recognized that Hawaii is not the only oceanic archipelago that has a very clear linear chronosequence of islands. This was expanded by Carrie Shaw and Rosemary Gillespie in 2016. You can see here the kind of linear regressions are all quite tight, not just for Hawaii, but for the Societies, the Australs, the Marquesas, the Galapagos, the Canaries, and the Azores. So if Warren Wagner and Vicky Funk are right about this, that evolution should be to some degree predictable based on the geologic history of Hawaii. It may also be the case that it's predictable in all these other archipelagos throughout the Pacific and the Atlantic. So what do we find? All of these animal lineages in Hawaii and all of the studies that I've cited below all show a very clear progression rule for Hawaiian animals. There's a few exceptions to this, but the predominant pattern is that animals in Hawaii show progression rule. Okay, I'm going to switch gears now, talk about another kind of possibility of predictability in evolution, something called the island syndrome. The island syndrome has its roots in both Darwin and Wallace's work, like many things in biology, but was really formalized by Sherwin Carlquist in the 1950s and 1960s. And we're probably familiar with some classic examples, gigantism and Galapagos tortoises, dwarfism in foxes, flightlessness in birds, melanism in lizards in the Greek islands, mega-herbs or the evolution of woodiness in island plants, seed gigantism in plants, etc., etc. The one that I want to dig into today is kind of an expansion of seed gigantism in plants, and that Carlquist proposed that not just mass, but other things evolve in a predictable way in terms of a loss of dispersal potential when plants make it to islands, particularly those that are distant from any mainland source. So the last concept that I want to touch on, predictability and evolution, is something called the taxon cycle. And this is work that really had its roots in paleontology, But E.O. Wilson really expanded it, thinking about ants on islands in the 1950s and 1960s again. And basically, the idea that he had was that ants, for example, in southeastern Asia, are primarily inner rainforest lineages, lots of diversity in the rainforest, but they sometimes occupy these what he called marginal habitats. I don't think it's quite the right term, but riparian habitats, intertidal habitats, coastal habitats, and that these were the lineages that were most likely in storm events to get swept out and raft to islands out into the Pacific. And then he was thinking about the near Pacific, what was then termed Melanesia, and that some lineages would make it out and ultimately go extinct. He had some other kind of key pieces to this taxon cycle. One was that if they were successful after kind of hitting a so-called beachhead, as he called them, in the Pacific, that you would typically see a habitat shift from these open marginal habitats like coastal habitats or riparian habitats in early stages to rainforests again. And then there would be diversification that would accompany these habitat shifts into the rainforest, and particularly upslope inland on these islands in Melanesia. He also thought that essentially this was a cul-de-sac or a one-way street where the cycle emphasized that islands are generally endpoints for evolution, where the species eventually, even after they diversify, they become very narrow endemics, both in terms of range and ecology as they adapt to island interiors, and that ultimately sets them up for the high extinction rates that we find on islands. However, one important point that I want to make is that it's often missed in his work, but it's very clearly there, and you can see it here, where he thought that in some cases you get reinitiation of the cycle, right? And the cycle here is that although island interior species often go extinct, they sometimes reinitiate this new phase of expansion through these so-called marginal habitats, starting the cycle over again. That is, essentially, you get derived species from island interior species occupying coastal habitats, riparian habitats again, and then setting up for dispersal and the taxon cycle starting over once again. Okay, so just to put some microbes in the talk, but I think that they're relevant. Some of the work that I did with Tad Fukami as well as Samantha Ford was using an experimental microbial system, Pseudomonas fluorescens, that rapidly diversifies through the process of adaptive radiation in experimental microcosms to test some of these ideas that we've been thinking about. And not to get into the nitty gritty, but in this, we basically developed the hypothesis that accounting for the prior evolutionary history, that is the traits that the species bring to the islands that they arrive on, as well as the immigration history, that is the timing and order of colonization, can largely determine the extent of diversification. And that this may be applicable to the otherwise puzzling variation in the extent of diversification, why some lineages diversify like crazy and other lineages diversify little or not at all after landing on islands. And that getting at the prior evolutionary history as well as the immigration history may be in part possible due to advances in phylogenetics. So, moving to the second part of the talk, general patterns of endemism in island plants and the lack of genetic diversity in Hawaiian plants. This is work that I did with Jeff Stallman, who at the time was a master's student at UH-Hilo, working with Don Hemes, myself, and others, and is now the fungal conservation coordinator. I think that's the coolest thing that this position exists at the Indianapolis Zoo, along with undergraduate Kira Robinson, who's the Don Hemes Award winner and now a master's student in mycology at Cal State East Bay. And what we did in this is we used updated checklists where we could get comparable data from all of these islands and archipelagos for all these taxonomic groups. So the Geomycetes, I don't even tell this group, are the mushroom-forming fungi. Lichens in the kind of yellow, kind of orangey color, the ferns and the blue, angiosperms in pink, and bryophytes in green. And probably everybody's already noticed and is not surprised by Hawaii, right? Endemism in Hawaii is exceptionally high. Endemism for mushroom-forming fungi is about 80%. And while we expect these numbers to change a little bit as species discovery continues over coming decades, we thought it would be good to get this out there as a starting point and kind of stimulate this discussion in the literature. One of the things that you've probably also noticed is that angiosperms, flowering plants, are the great speciators on islands and archipelagos around the world. Okay, so speaking of, Hawaiian plants are just staggeringly beautiful. It's really an incredible honor in my life to be able to work on these lineages. I'm very excited every day that I get to do this. These are just, some examples that I've pulled of many, and I apologize if your favorite plants aren't on this slide. But one of the things I want to emphasize with this slide is that all of these lineages have an incredible amount of ecological and morphological diversity amongst the species in Hawaii, and most all of their species are endemic to Hawaii. However, we see in all of the published studies that I cited here, very little genetic diversity both within and among species. And this is what I like to call the Hawaiian plant paradox. How is it that we get this extreme ecomorphological differentiation, lots and lots of species despite limited genetic differentiation? And I hopefully will help answer that question with the rest of the talk. This is not a conservation talk, but I would be remiss if I didn't mention that 108 species of the native Hawaiian flora are already recently extinct since European contact, and another 257, this is greater than a quarter of all species, have less than 50 individuals remaining in the wild. So these are critically endangered species. Okay, so to kind of further illustrate the point of the lack of genetic diversity in Hawaiian plants, again, Jeff Stallman, myself and others did a DNA barcoding project of native Hawaiian plant lineages. We looked at 385 species and 20 native lineages, and we used the 4 most commonly used barcodes as well as the concatenated set for some of those lineages. And you can see here at best, regardless of what locus or the concatenated set, we get about 22% accurate identification based on DNA barcodes. There just isn't the genetic diversity that there is in animals. Animals tend to be, about 95% for DNA barcodes, and we just can't use them yet based on current methodology in Hawaii. Okay, recently a bunch of us got together, an effort led by Jose Cerca, to write up a review about, well, okay, what do we know about the genomics of oceanic island radiations? And are we even ready to write a review? One of the things that I thought was illustrative that came out of this exercise is that if we look at the number of well-documented, that is, peer-reviewed, published papers on adaptive radiations across taxonomic groups, we find 119 different adaptive radiations in the literature. And you see here, probably already noticed that it's really arthropods and it's plants that are doing the heavy lifting in terms of adaptive radiation on islands. And then within plants, it's the Asteraceae, or this daisy family. It seems to be particularly good at going through adaptive radiation once they make it to oceanic islands. Classic examples of adaptive radiation from the Hawaiian flora are Hawaiian bidens, which I'm going to focus on mostly for the rest of the talk. But then I'm going to talk about our new work on the Hawaiian Silver Sword Alliance. And I'm not going to talk today about our work on Hawaiian tetramalnopium, but it's another adaptive radiation that's really interesting. I just don't have time to get into it today. So progress in terms of phylogenomic methods, and are we just crazy to work on these kinds of lineages that have such little genetic diversity? Well, first I want to back up a little bit and talk about progress in terms of phylogenomics for understanding the evolution of Asteraceae as a whole. This is really brilliant work done by Jen Mandel, University of Memphis, that we've piggybacked on here, and we're collaborating with her. And this is a big phylogeny for the Asteraceae, focusing on the subfamily and tribe level. And we find, based on hive-seek, or sometimes called target capture, phylogenomic approach. And essentially, if you're not familiar with this, this is basically a couple hundred to a couple thousand loci at a time being sequenced, usually about 500 to 1,000 base pairs each, so somewhere on the order of about 500,000 to 1 million base pairs per individual, per probe set or target capture set. And when we do this, we find all traditionally taxonomically described subfamilies and tribes in the Asteraceae are monophyletic. This is an extraordinary thing where the phylogenomics matches perfectly with the traditional taxonomy. We also, in terms of time-calibrated phylogeny, see that really the explosion of diversification is following the end Cretaceous mass extinction event, and we really get going in the Eocene and Oligocene. And that this resulted in the most diverse plant family on Earth, with about 32,000 described species and counting, extant species and counting. But we recently investigated diversity across plant families on all islands across the world that have checklist data. And it's not the Asteraceae, it's the second most diverse family globally that's the most diverse, both in terms of endemic diversity as well as native species diversity on islands, the Orchidaceae, followed by the Rubiaceae, this is the coffee family, and then the Asteraceae. But if we restrict our data set just to oceanic islands, we move that around, and the asters are both have the greatest number of endemic species as well as the greatest number of native species on oceanic islands like Hawaii and the others that I mentioned. Why is this? So Asteraceae, they possess a unique fruit morphology that I'm going to dig into in gory detail in a moment, that aids in long distance dispersal. So we think they're really good at getting out to oceanic islands and then spreading amongst islands. They have a kind of head-like inflorescence that's called a capitulum that often attracts generalist pollinators. So this is thought to be good for establishment on new islands. And many species are capable of selfing, again, also thought to be an advantage when dispersing to a new island or archipelago. So I want to focus in on these two lineages, primarily the Bidens, and I'll come back to the silver swords. But I just want to mention Jerry Carr, who is in the botany department here for many years, dubbed the Hawaiian Bidens and the Silver Sword Alliance masters of adaptive radiation. He thought that these two lineages were the premier examples of adaptive radiation in plants in Hawaii and possibly the world. And adaptive radiation can be defined as the rapid diversification of a single evolutionary lineage into a large number of descendant species adapted to many ecological roles. And we know from Bruce Baldwin and Michael Sanderson's work, and then subsequent work by Michael Landis and others, that the Hawaiian Silver Sword Alliance arrived in Hawaii just about the time that Kauai was where the Big Island is now, and very clearly follows progression rule and walked down the island chain as suitable habitat on Oahu became available, the islands of Maui Nui and the Big Island. And we've found that colonization of the Hawaiian Bidens is much younger, about 1.8 million years, so just about the time that Maui Nui is breaking the surface. Kauai and Oahu are already high islands, but long before the formation of Hawaii Island. So I want to ask you guys to think about this. Do we expect progression rule for late arriving lineages? And Hawaiian Bidens is just one of many that have come much more recently than the full age of the main Hawaiian Islands. And what would it look like if we were to set up an expectation in our minds for progression rule for late arriving lineages? Okay, before I do that and show the evidence, I just want to introduce you a bit to this lineage. The Hawaiian Bidens, or the Ko'oko'olau, have this incredible diversity in terms of their kind of ecomorphology. There's succulent types, there's trees in the Waianaes, there's lianas in the Alakai Swamp, there's lava flow specialists, et cetera, et cetera. There are 20 endemic species. They're all hexaploid. While continental Bidens have variable ploidy levels, they can be diploid, tetraploid, or hexaploid. There's actually greater morphological and ecological variation in these 20 species in Hawaii than there is in the remaining 200 species that are found on 5 continents around the globe. We and others before us have grown them in controlled common garden experiments, and we get the phenotypes back exactly as we find them in the wild. So this is not a result of phenotypic plasticity. These are genetic differences amongst these species. And they tend to be single island endemic. About 70% of species are on a single island. Within islands, they tend to habitat segregate. And if you include this species, Bidens cosmoides, it has this radically different floral architecture and it's thought to be bird pollinated, while all the others are thought to be insect pollinated. 93% of species are genetically isolated from one another. However, when they do come into contact, they readily hybridize, and hybrid swarms form at regions of range overlap. About half of them are of pressing conservation concern, threatened, endangered, critically endangered. All are of high cultural and medical importance. And just to illustrate that point, this is a brochure from the med school here about the koko lau, and referring to the la'au, lapa'au, or the traditional Hawaiian use of this plant. It's recommended as a tonic to treat throat and stomach ailments, severe asthma, etc. Here's the really extraordinary thing. Cultures on those 5 continents around the world have converged on the same medicinal use of the Bidens plants in 5 different continents. Okay, so it's not all good news. There's 2 recently introduced Bidens to Hawaii. These are continental species with a center of origin in around Mexico. Bidens pilosa and Bidens alba have become significant invasive species in Hawaii, documented early by Kurt Daler to cause significant ecological harm. And we were interested in the possibility of these invasive species hybridizing with the native species. They sometimes are found right next to each other. So for example, if you go on the Manoa Cliffs Trail, you'll see Bidens pilosa growing right next to Bidens asymmetrica. And we found, luckily, that we weren't able to produce any crosses between the invasives and the natives, at least using the methodology that we used here in the greenhouses, but that using that exact same methodology, we could make viable fertile offspring amongst all possible combinations of the Hawaiians that we tested. While this study was motivated by conservation, I think it tells us something interesting about evolution, right? That this 1.8 million year time frame in Hawaii is insufficient for complete genetic isolation among the Hawaiian endemics, and it hints that large structural macromutations are not responsible for the diversity of the adaptive radiation, as we suspect in the Hawaiian Silver Sword Alliance, which I'll get to in a little bit. What's our time? Half hour? Okay, perfect. So progress in phylogenetic and phylogenomic now methods to reconstruct the evolutionary history of this recent rapid diversification event. So we have now, over the last 20 years, collected 134 species from throughout Polynesia, around the Pacific Rim, and elsewhere in the world. We've vouchered them all here at Rock, at NTBG, at Bishop, and the Smithsonian. We've kind of followed the technologies, tried to keep up with the train as it's sped down faster and faster down the tracks. We started with Sanger sequencing and then moved to Illumina high-seq shotgun sequencing. Most recently, we're using the PacBio Smart Cell, long read, high fidelity, and high-C optical mapping. They all have these fancy names. But I'm going to talk a little bit about what we get out of each of these technologies. This is our original effort. So Sanger sequencing, we use 4 plastid or chloroplast markers and 2 nuclear markers that are traditionally used for species level resolution of molecular phylogenies in plants all over the world. And they fail miserably in the Hawaiian Bidens. This is this ITS, but the trees for all of the 5 markers are essentially identical. I'll walk you through a few important things. This is the node that unites all of the Hawaiian species in red, plus at the time, these 2 undescribed species in the Marquesas. And we find strong statistical support for the monophyly of the Hawaiian plus Marquesan taxa. I'll come back to the Marquesas in a little bit. We find, based on this taxonomic sampling of 35 species, that the Bidens alba and pilosa I just mentioned, those invasive species now in Hawaii, are in the clade that is likely the most recent common ancestor of the Hawaiian plus Marquesan group. And I'll come back to that as well. And we find essentially 0 genetic diversity amongst the Hawaiian species at this marker, ITS, and these others that are so good at resolving molecular phylogenies in plants elsewhere. We find that at the most, we have less than 1% genetic distance between these Hawaiian species that I just showed you are so incredibly ecologically and morphologically different from each other. When we get out to these African outgroups, we see that we've got about 16% divergence, and this is, it's clock-like evolution. It's not that IPS isn't accumulating mutations. It's just that this is among the fastest diversification events ever documented in plants, particularly so on a per unit area basis. Okay, so now this is moving into the shotgun sequencing approach. So when I took the position at UH-Hilo, we decided, okay, let's go for this new, Illumina approach. And doing this, we now get a fully bifurcating, generally well-supported phylogenetic hypothesis for the Hawaiian bidens. I'll just draw a box around the Hawaiian species, and I'll just blow up the statistical support for the monophyly of the Hawaiian species. And it's as strong as it can be, both from a Bayesian as well as a likelihood approach. I don't have time to get into the details here, but there's lots of interesting things that I wish we could talk about, but I'll come back to that tree in just a moment. But now I want to come back to progression rule, right? So as I mentioned, the silver sword arrived about 5 million years ago when Kauai was the big island is. Clearly walked down the island chain with progression rule, but our estimates, multiple approaches, we all kind of converged and multiple publications on about 1.8 million years, just about the time the geology tells us Maui Nui is forming. And so what do we find in terms of progression rule? What I'm going to show you now is just the exact same cladogram that I showed you, but just for the Hawaiian species. This is the Illumina data, and I'm going to walk you through it because phylogenetic trees are always complicated. But if you look right here in green, if you look at the color coding, this is Maui Nui, and you see colonization of Maui Nui from uncertain ancestry. But after colonization of Maui Nui, subsequent in situ speciation and buildup of Maui Nui endemics, and then from Maui Nui, dispersal, and then subsequent speciation and buildup of endemic diversity on Hawaii Island, both exactly, almost perfectly coincident with when we expect suitable habitat to be available on both of those islands or Maui Nui in terms of the complex. I think that this is very clear evidence for progression rule for a late arriving lineage coming in the middle of the formation of the geologic history of the main Hawaiian Islands. But now if we look up here, these are purple, Kauai, and orange, Oahu, and a little sprinkling of other things. But basically, it's a big mix and a mess. And I think this is exactly what we should expect. Kauai and Oahu were already here as high islands when the lineage arrived in Hawaii. So there's no waiting time for that island to become available. We shouldn't expect progression rule for those older islands that are older than the estimate of the time of colonization for the lineage in Hawaii. And that's exactly what we find. Okay. Now I want to talk a little bit about broader work that we've been doing throughout the Pacific. I had a great opportunity to work with Vicki Funk and Sherwin Carlquist, in addition to many other leading botanists. And this was a kind of 25 year field collecting effort throughout the Pacific. What comes out of this is that the Biden's radiation is not just in Hawaii, but it's also in other areas of Polynesia. It's pretty much restricted to Polynesia. The Marquesas, these are just some representative photographs. The Society Islands, and all the way down to the deep South Pacific and the Austral Islands. There's 42 currently described species, 20 in Hawaii and 22 in other Polynesian islands. So what do we get? So this is based on the Sanger sequencing approach, but this is all 6 of those loci that I mentioned together. And now we have much better, taxonomic representation. This is the Pacific, the Polynesian group, and then these are various outgroups kind of walking around the Pacific Rim and elsewhere. And what do we find? Well, based on this greater taxonomic sampling, we've moved the most recent common ancestor from Central America to South America, but with weak statistical support. So we're still not sure, and we're working on this right now. We see a single colonization of the Pacific, but we can't parse Hawaii from the Marquesas, and it was probably so quick, coincident in time, the dispersal between the two, that we're not sure if they first arrived in Hawaii or first arrived in Marquesas, and we're hopefully going to be able to answer that real soon too. But Hawaii is clearly monophyletic adaptive radiation. And then the Marquesas give rise to the Society Islands radiation. once, but then also a second natural colonization from the Marquesas to the Society Islands, and then from the Society Islands down to Hawai'i and the Austral Islands. Okay, what we're doing now is for those 134 species now, this tree is actually cut off. We had more in this, but we have now the 134 species. We're doing this hive-seq phylogenomics, which is what we should have done in 2016, and not gone with the Illumina shotgun sequencing approach. And we're using all 3 probe sets that are available for working on the Asteraceae. So we're going to, hopefully, we've done a preliminary run, and we're getting about 2 million base pairs per individual and kind of slam dunk phylogenetic resolution. They're not quite ready for me to present, but hopefully soon we'll answer some of these questions about the historical biogeography with good confidence. So along the way, we're discovering new species and describing those species. So this was a pandemic project. I ended up back home on Kauai for 2 years during the pandemic, and I thought, okay, I better do something or I'm going to lose my mind. So one of the projects that I started with was Ken Wood to describe a new Biden species that he and others at NTBG had discovered in the blue hole. That's basically right below Mount Waialeale, the wettest spot on the planet. And these plants, you can't see them here, but they kind of cling to the side of the what's called the wall of tears on the interior of the blue hole, as well as they're found at the base of the thundering waterfall at the back of Limahuli Valley on the north shore of Kauai. Unfortunately, there's only about 750 individuals left. So we did a IUCN Red List workup on them. 3 declining populations. It turns out, that they're critically endangered based on the IUCN. The threats are the typical cast of characters for Hawaiian plants: feral pigs, rats, slugs, invasive plants, and we lost the population to a landslide in the 2018 mega flood on the north shore of Kauai when 49 inches of rain came down in 24 hours. So they're very vulnerable. The good news is that we have conservation cultivations growing at NTBG. And I just want to mention a couple others that are in progress. One from the Society Islands, Huahinensis, and one from Maui, Lihauensis, hopefully coming soon. And my guess is that more will continue to come out of our work as we are out in the field. Okay, so moving into the kind of ecology of dispersal trait evolution. I thought this picture was really cool. This picture is taken in Wailua on Kauai, and this is a barn owl. It's not a puea. This is an introduced owl. But what we see right here are the achenes, or the dry fruits of the bidens, stuck to the feathers of this barn owl. And because I live just down the road from Luke Evslin, I know that almost with full certainty, because they're everywhere, that this is Bidens pilosa, this introduced invasive Bidens. And this is the dry fruits, right? And this is how they get around. They have these barbed awns, they're called, on the end. They're essentially like porcupine quills. They get readily caught in bird feathers, as well as mammal fur, and your socks and shoelaces as well. But this is how they're thought to have gotten out into the Pacific to begin with, presumably attached to bird feathers, likely seabird or shorebird feathers. Okay, so Shoreham Carlquist used this lineage as his kind of illustration of what he thought was the best example of the loss of dispersal potential and island plants, and that he hypothesized that the most recent common ancestor of the Polynesian Bidens looked something like this, something like Bidens pilosa, which is kind of typical for continental species in the genus. And he hypothesized that island interior species will tend to have vestigial awns, or the awns absent. This whole structure will be reduced or lost altogether. He hypothesized that island interior species will have these hairs that are what we call retros hairs. They go the other way than the barbs to make sure that you really get lodged in, that they'll be short and stiff or lost altogether, or what's termed glabrous if the achene is smooth. That island interior species will tend to evolve irregular shapes, such as arcs and helices for local scale dispersal only. And the island interior species are going to get bigger and have more mass. That In addition, they'll be longer and wider. So this is kind of the hypothesis that he set up in the 1960s, but he had no quantitative data. And so we decided, this is another pandemic project of mine, back on Kauai at NTBT. I said, well, okay, why don't we actually go after this and add some quantitative data and analyze it in a phylogenetically explicit way. So these are images of the achenes in our study. So up here at the top, I know it's a little small on the screen, are phytons alba and pilosa. You can see those barbed awns. And then we get into the endemic Hawaiian species. We have these two coastal species that look pretty darn similar to the continental species. They have maybe a little bit of reduction of the awns and the hairiness and the barbs, but generally they look like they're well adapted for long distance dispersal. I'll come back to that. And then we have these species that are kind of mixed. They can go from the coast on up, interior, up in elevation, and you kind of get a diversity of forms, including one that's winged and seems to glide away from the parent plant, especially on the sea cliffs where it's found, Bidens maliensis. And then you get these purely upland island interior species. And I think, if you kind of look at it, I think you can kind of start to see that, yeah, we're losing, and we get reduction or complete loss of the awns. We get these smooth, glabrous achenes that have lost their hairs. We get these helical, twisted shapes like Bidens torta here in the Waianaes on Oahu, etc. So we used a kind of new updated phylogenomic hypothesis based on the Illumina shotgun sequencing approach. And essentially, this is a complicated figure, this kind of rainbow figure, but here's the tree. The letters indicate the species. We have almost all of them. And I'm just showing you the predictor variables in the kind of yellow to green hues and the response variables in the blue hues that are statistically significantly related to each other. All the other ones I've just omitted for simplicity. So we have three response variables: achene length, the presence or absence of awns, and whether they're sectos or glabros that are statistically significantly related to these various predictors, for example, elevation minimum, elevation range, etc. And I'll get into that in just a little more detail in a second. One of the things I want to point out is those two coastal species that I showed you that look a lot like the continental presumed ancestor that still have those barbed awns and those hairs. They're not basal species like you would expect from the taxon cycle. These are highly derived species and they're not sister species, right? So I think what we're seeing here is that these endemic Hawaiian coastal species are essentially what E.O. Wilson predicted from the taxon cycle, that they're this phase, this reemergence of what he called marginal habitat derived species, and they're breaking this one-way street towards extinction in island interiors. That Bidens are resetting the stage by evolving new coastal species that have what appears to be the adaptations necessary for dispersal off island onto other islands and archipelagos. Okay, just to summarize, in terms of Carlquist's hypotheses, we find statistically significant relationships for the loss of awns, for the loss of hairs, but we don't find significance for irregular shapes such as arcs and helices, and we don't find increase in mass, but we do find increase in length, but not width. So it's kind of equivocal there. Okay, moving into the last parts of the talk here. I think timing's fine. Reference genomes and comparative genomics. So this is more recent work that was really spearheaded in my lab by Dr. Renee Bellinger at UH Hilo, who at the time was a postdoc with us, who now runs the Conservation Genomics Facility for the USGS up in Volcano. And what we did is we used the PacBio high sequencing approach to develop a reference genome. As far as I'm aware, this is the first reference genome for any Hawaiian plant adaptive radiation. And it's massive. It's 7 billion base pairs. It's this hexaploid, giant genome. We have 55 billion base pairs of data. This in purple here is single copy, in teal duplicated, green fragmented, and pink incomplete. So we have no fragmentation and we have no incomplete parts of the genome. So it's a high quality genome, well beyond the threshold for model organism reference genome. We see it's largely duplicated as we would expect for a hexaploid genome, and we compare it to these other asters. You'll notice that most of the asters that have high quality reference genomes come from agriculturally important species. If we then expand it to other hexaploid plant genomes, we see pretty much the exact same thing. Very high percentage of the genome is duplicated, as you would expect, and then some like strawberry that are octoploid are just incredibly difficult to work with and need more passes. So a highly complete genome, a high proportion duplicated as we would expect, but this part, I'm not going to show you the evidence, I'm just going to tell you, very high proportion of transposable elements. We find that it's almost the exact same percentage of transposable elements that we find in the hollow genome. And my suspicion, and this is just a hypothesis, is that this is going to be an important part of the story about evolvability in Hawaii as well as elsewhere as we move forward. Okay, so speaking of that, a path forward. We were fortunate recently to get NSF funding, and I'm collaborating with Daniel Jones, who's an evolutionary developmental genomicist at Clemson, who really is interested in the evolution of the capitulum. Is his whole lab, his whole career is focused on how do we get this incredible diversity in this most diverse plant family on the planet. And he has all these tools in his toolkit, including the functional genomics and many other things, to be able to kind of track the development and pinpoint the genes responsible for the diversity of the traits that we see in the Hawaiian radiation as well as continental bidens. And then in relation to the habitats that these species are found, we're working on the kind of ecophysiology in collaboration with Chris Muir, formerly here of UH Manoa Botany and now at University of Wisconsin-Madison, to do experimental work as well as observational work in the field to get at the ecophysiological adaptations. And you can just see here a little kind of taste of the kind of stomatal differences on upper and lower surfaces for continental. The different Hawaiian species that are in different habitats have very predictable differences in terms of the way they structure their stomata, et cetera, et cetera. Very excited about this. Really a wonderful group of people to work with, and hopefully the next time I have the opportunity to talk with you guys, we'll have some more exciting results to share. The last thing I'm going to say about the path forward is that in moving over here a year and a half ago, we really started to put a lot of energy into working on the Hawaiian silver sword adaptive radiation. Many of you, I think, are well familiar. But 34 endemic species, they naturally hybridize like crazy. There's 46 documented natural hybrid taxa. Unfortunately, they're not doing well, whether it's the IUCN Red List or the U.S. Fish and Wildlife. Of the species that have been assessed, all of them are either, there's 2 that are extinct, that says 1, but it's 2, 17 that are critically endangered, 8 that are endangered, 2 that are vulnerable, and the rest just haven't been assessed yet. We're getting reports from the field botanists all across Hawaii. Populations are crashing. It's an extinction crisis, and if we don't do anything about it, we're going to lose this iconic radiation in Hawaii. So with that in mind, we've kind of jumped in both in terms of evolutionary genomics, but also conservation genomics. If you're interested in the conservation genomics side of things, master's student Emma P.E. Anaya, who master's student in the botany department with me, is giving her proposal seminar this Friday in St. John at 11:45, and she's going to talk about her work on developing a reference genome and transcriptome for the critically endangered Haleakala silver sword. The other thing that we're doing as a start is there's 8 known large structural macula mutations, or whatever, these 8 known genome types based on Jerry Carr work here doing chromosomal smashes in the 1980s. And we're going over those using this PacBio technology to get these high quality reference genomes. We just have our first in draft form. And in addition, we're making sure that we get one of each of the different chromosome numbers. They've lost a chromosome pair, the ancestral conditions N equals 14, and then some of the derived species on Big Island especially, but also here on Oahu are N equals 13. Some other goals of the project, we have this wonderful team of people. We're kind of building support for the project, and we're hoping to continue to like I said at the beginning, kind of get real wind under our wings, and excited to collaborate with the INA Informatics Network. Dr. Yvonne Chan and Dr. Ethan Hunt at Iolani Schools have this incredible program. If you're not aware of it, it's like 26 partner high schools throughout the state, public and private, where the high school students are sequencing genomes. They're sequencing right now the exact same genome that we have, the draft genome, Debadia plantagenea, alongside the PacBio They're using Oxford nanopore technology, and we're just asking the question with these high school students, do we get the exact same genome back? Using two different, exact same individual plant, exact same DNA extraction, we're gonna see if we get the exact same thing, and I'm down there next week excited to work with those high school students. Okay, so one take-home message here, I think, is that evolution on islands may be largely predictable, but that these insights are really only made possible by the combination of fields that are generally viewed as disparate, things like genomics, geology, ecology. And then if I would just add to this, I think we're just at an incredibly exciting time to be island biologists, and the technologies that are available to us, the kinds of questions that we can ask and hopefully answer that weren't accessible to Sherman Carlquist, Leo Wilson, and others in the 1950s and 1960s. I just wanted to say many mahalos to my lab group at UH-Hilo, sources of funding there, and many mahalos to my lab group as it's developing here at UH Manila and sources of funding. And with that, I'll take any questions.